Quote, vitamin means that one has to eat it. What one has to eat is the first concern of the chef, not the scientist.
Oh, I love that one. That's Albert Szent-Györgyi.
Yeah, the very guy who helped discover vitamin C. And it just perfectly captures this, I don't know, this weird liminal space that vitamins occupy in our culture today.
Right, because they're caught somewhere between like culinary advice and over-the-counter wellness marketing.
Exactly, but they're actually hardcore biochemistry. And for you listening right now, whether you're, a medical student prepping for clinical rotations or maybe a young professional studying for your boards or just a dedicated learner, we are completely throwing out the wellness buzzwords today.
Yeah, we're taking the scientist's view.
We are. We're doing a deep dive straight into a foundational text, which is The Vitamins: Fundamental Aspects in Nutrition and Health, the third edition by Gerald F. Combs, Jr. The mission here is to equip you with a concrete clinical framework for understanding how these compounds actually work, like mechanistically in the human body.
Because when you strip away the marketing hype, it reveals a deeply complex biological reality. The textbook gives us this rigorous scientific baseline that honestly, it fundamentally shifts how you view nutrition in a clinical setting.
Oh, absolutely.
We're going to look at the clear physiological mechanisms, the chemical properties that dictate how the body handles these molecules, and this is key, the clinical realities of what happens when the whole system breaks down.
Right. So by the end of this deep dive, you're going to have a mechanistic understanding of pathogenesis, not just, a memorized list of symptoms on a flashcard.
Exactly. But to truly understand how vitamins impact health, we have to start by defining what a vitamin actually is. Because the scientific definition Combs provides is, it's much stricter than the popular idea of just healthy food.
Much stricter. According to the text, a compound has to meet 5 very specific criteria to even earn the title of a vitamin.
Right, 5 criteria.
First, it's got to be an organic compound that is distinctly different from fats, carbs, and proteins.
You have macros.
Right. Second, it has to be a natural component of foods, usually present in just minute amounts. Third, it is essential for normal physiological function, so maintenance, growth, development.
Which is a big one.
Huge. Fourth, its absence or underutilization must cause a specific deficiency syndrome. And fifth, and this is the fascinating part, it cannot be synthesized by the host in amounts adequate to meet normal physiological needs.
And that 5th criteria, the whole inability of the host to synthesize the compound, is where we see that vitamin is not just a chemical classification. It's actually an evolutionary relationship.
Wait, expand on that. An evolutionary relationship?
Yeah, so the textbook highlights vitamin C to illustrate this. For humans, and interestingly enough, for guinea pigs.
Guinea pigs.
Yeah, humans and guinea pigs. For us, vitamin C is an essential vitamin because our species lack a specific enzyme that's required to synthesize it from glucose. So we have to ingest it.
Okay, wow.
But for a cow or a dog or a sheep, ascorbic acid is not a vitamin at all. It's simply another metabolic intermediate that their own livers manufacture in perfectly adequate amounts.
That's wild. So it's just an evolutionary quirk.
Yeah.
The chemical structure of ascorbic acid is identical, whether it's in a human or a cow, but its classification as a vitamin is entirely dependent on the species.
Exactly.
It brings to mind the macronutrients. Fats, carbs, proteins. If we think of those as like the fuel, the chassis, and the engine block of a car, I've always thought of vitamins as the tiny little spark plugs.
Oh, that's a great analogy.
Right. Because you don't need a large physical volume of them, but they're these highly specific, highly complex components that force vital chemical reactions to happen. Without them, the engine's exhaust turns toxic, and the whole metabolic machine just grinds to a halt.
And realizing that the absence of a tiny unseen factor could cause massive physiological dysfunction that actually sparked a scientific revolution.
Because before that, everything was germ theory.
It's exactly.
Yeah.
In the late 19th and early 20th centuries, the medical establishment was entirely dominated by the germ theory of disease. Science was actively hunting for pathogens, microbes, environmental toxins.
They were looking for the bad guy.
Right. The prevailing logic was that disease was caused by the presence of a hostile agent. So introducing the concept of a deficiency disease, proposing that morbidity was caused by something that wasn't there, that required a massive paradigm shift in clinical diagnostics.
Which means the pathology of a deficiency disease doesn't just happen overnight. You don't skip an orange on Tuesday and present with full-blown scurvy on Wednesday.
No, thankfully not.
There is a very specific timeline, which the text calls the cascade of effects. And mastering this cascade seems absolutely fundamental for anyone in clinical assessment because it maps the exact sequence of how a system failed.
It does. It maps it out perfectly.
It begins with inadequate intake or absorption, which then fails to support the body's tissue stores. As those stores deplete, they can no longer support normal metabolic turnover. And that leads to what the book termed a biochemical lesion.
Right. And altered cellular metabolism is the essence of that biochemical lesion. The enzymes that rely on the vitamin simply stop functioning, causing a bottleneck in a very specific metabolic pathway.
But wait, let me push back on this a bit. Because if the very first thing that goes wrong is an invisible biochemical lesion, just a microscopic failure in cellular metabolism, how are doctors supposed to catch it? We're essentially flying blind if we wait for a patient to complain of physical symptoms.
Well, that's the thing. Only after that invisible lesion is firmly established do you see an impairment in actual physiological function. And only after the physiological function is impaired do you get the classic clinical signs and symptoms.
Which is too late in some cases.
Yeah, because if the deficiency is allowed to progress further, you see morphological changes. Actual structural, irreversible damage to the tissues, which ultimately leads to morbidity and mortality.
Right. So by the time a doctor sees overt morphological changes, like, say, the severe bone malformations and rickets caused by vitamin D deficiency, the structural damage is permanent. We have to catch the failure at the biochemical stage long before that structural damage occurs.
And modern clinical assessment relies almost entirely on that exact logic. The physical exam is secondary to biochemical indicators. Thiamine, or vitamin B1, is a perfect historical example of this.
Oh, beriberi.
Yeah. The classic clinical manifestation of severe thiamine deficiency is beriberi. It presents with anorexia, profound weight loss, insomnia, hyperirritability, and eventually cardiovascular or neurological collapse.
Just devastating.
It is. And historically, physicians had to wait for those devastating symptoms to appear to make a diagnosis. But today, modern diagnostics focus on testing specific blood and tissue enzyme levels. So for thiamine, a clinician will measure erythrocyte transcatalyase activity in the blood.
Because transcatalyase needs thiamine to work.
Exactly. Because it requires thiamine to function, a drop in its activity reveals the biochemical lesion weeks or months before the patient experiences any neurological damage.
That's incredible. But to even know which metabolic pathway to test, you have to understand the physical chemistry of the vitamin in question.
You really do. It all comes back to chemistry.
And the textbook draws a hard line dividing all these compounds into two major camps based on a single physical property, which is solubility. It categorizes them into water-soluble vitamins and fat-soluble vitamins.
Which is so much more than just a convenient way to group them on a flashcard.
Oh, for sure. That single physical property dictates the entire life cycle of the molecule in the human body. How it's absorbed in the gut, how it travels through the blood, where it's stored, and how easily it becomes toxic.
Let's break that down. The water-soluble vitamins, which encompass the B-complex group and vitamin C, they possess polar or ionizable functional groups. So we were talking about carboxyl, keto, hydroxyl, or amino groups.
And blood is mostly water?
Exactly. Because human blood is primarily water, these polar molecules can be absorbed directly across the intestinal barrier and right into the portal vein. They dissolve easily, they travel freely.
But the fat-soluble ones are totally different.
Completely. The fat-soluble vitamins A, D, E, and K are entirely different beasts. Their chemical structures are predominantly characterized by aromatic and aliphatic rings and chains. They are lipophilic, meaning they repel water.
And because they repel water, they can't just dissolve into the bloodstream. Their enteric absorption requires this incredibly complex biological workaround.
Yeah, the body has to work for it.
The body has to secrete bile salts to emulsify them into my cells. Literally just like dish soap breaking up grease in a frying pan. And even then, they don't go straight to the blood.
No, they have to be packaged into calomicrons.
Which are basically microscopic lipid shuttles. And they have to travel through the lymphatic system before they're finally dumped into the venous circulation.
And that mechanical difference in absorption directly translates to how they are stored and how they're excreted. The body constantly filters water-soluble vitamins through the kidneys, excreting the excess in urine, so you generally have to replenish them frequently.
Right.
Fat-soluble vitamins, however, are retained. Because of their lipophilic nature, they're efficiently stored in the liver and in adipose tissue. The body literally hoards them.
Which fundamentally changes the clinical risk profile. I like to compare water-soluble vitamins to water in a leaky bucket. You have to keep topping it up constantly, but it's nearly impossible to overfill it because the excess just spills out.
That's a good way to look at it.
But fat-soluble vitamins act like a sealed rain barrel. They hold water incredibly well to get the body through a physiological drought. But if a patient decides to take massive pharmacologic doses of a fat-soluble vitamin over the counter, they are pumping a high-pressure fire hose into a sealed barrel.
And the barrel eventually bursts.
Exactly.
The medical term for that bursting is hypervitaminosis, and the textbook uses this to illustrate a fundamental pharmacological concept: the biphasic dose-response curve.
Oh, Paracelsus, right.
Yes, the 16th century physician Paracelsus famously observed that the dosage makes a thing either a poison or a remedy. At the lower end of the curve, increasing the dose of a vitamin cures a deficiency and restores health. But once you cross a specific threshold, continuing to increase the dose of a fat-soluble vitamin pushes the patient into severe toxicity.
Which is terrifying when you think about how many people just pop supplements. If a patient takes massive doses of vitamin D, assuming it will just, I don't know, boost their immune system or build stronger bones, that biological function goes into overdrive.
It does. Vitamin D promotes calcium absorption.
Right, so at toxic levels, the patient develops hypercalcemia, dangerously high levels of calcium just flooding the blood, and that excess mineral has to go somewhere. We aren't just talking about kidney stones here.
No, it's much worse.
We are talking about calcinosis, the actual calcification of soft tissues. The body literally begins depositing bone material into the kidneys, the lungs, and the walls of the blood vessels.
The systemic damage from hypervitaminosis D is a stark reminder that these are potent, hormonally active molecules. They aren't just benign supplements.
Yeah, they're powerful.
And understanding this baseline, the chemical framework of solubility and the biological framework of the deficiency cascade, that allows us to look at the specific pathogenic mechanisms of individual vitamins.
Let's do that. Let's look at some clinical snapshots from the text, because this is where it gets really interesting. Let's start with vitamin A.
Okay, vitamin A. It's actually a generic descriptor for compounds exhibiting the biological activity of retinol, including vitamins like retinol and retinoic acid. It has two major physiological roles: RAT, forming visual pigments in the retina, and critically regulating epithelial cell differentiation.
And the mechanism behind that second role, epithelial cell differentiation, is where the biochemistry perfectly explains the clinical symptom. Retinoic acid enters the nucleus of the cell and acts as a signaling molecule. It binds to receptors that regulate gene transcription.
Right.
It literally tells the body's mucus-secreting cells to stay soft and wet. And without that specific chemical signal, the default genetic program of an epithelial cell kicks in, and it becomes tough, dry, and keratinized. Literally exactly like the skin on the back of your hand.
And the clinical manifestation of that biochemical failure is devastating. The conjunctiva of the eye relies on those mucus secretions to stay lubricated.
Right.
So in a state of severe vitamin A deficiency, the eye dries out. It's a condition called xerophthalmia. The epithelial tissue keratinizes, and the patient develops Bitot spots.
Oh, I've seen pictures of those.
Yeah, they are raised, foamy patches on the surface of the eye made entirely of sloughed-off keratin debris and actual bacillus bacteria growing on the compromised tissue. The clinical sign is a direct, unavoidable consequence of the altered gene transcription.
It's just a cascade of failure. Moving from the eyes to the skeleton, vitamin D operates on a similarly profound genetic level. Structurally, the text notes that vitamin D is a side chain substituted open ring steroid.
A steroid, exactly.
Its active form functions entirely as a hormone to maintain calcium homeostasis. It binds to receptors in the enterocytes of the gut to trigger the synthesis of calcium-binding proteins. So if a patient lacks vitamin D, they can drink all the milk they want. But without those binding proteins, the dietary calcium simply passes through the digestive tract unabsorbed.
And the skeletal system becomes the collateral damage of that failure. To maintain critical calcium levels in the blood for nerve signaling and muscle contraction, the body begins mobilizing calcium directly out of the skeleton.
It just steals it from the bones.
Exactly. In children, this demineralization results in rickets, where the growing bones become soft and literally bend under the weight of the body. In adults whose growth plates have fused, the condition is called osteomalacia, leading to deep bone pain and severe fracture risk.
Let's shift to vitamin E, because this provides an absolute masterclass in how physical chemistry dictates biological function. Vitamin E is highly lipophilic. It's a fat-soluble molecule with a long aliphatic tail.
Right.
Because of that structure, it doesn't just float around in the watery cytoplasm of the cell. It physically embeds itself directly into the hydrophobic core of the body's cellular membranes, sitting right alongside the polyunsaturated membrane phospholipids.
And those polyunsaturated lipids are highly susceptible to oxidative damage. When metabolic free radicals attack the cell membrane, they trigger a destructive chain reaction known as lipid peroxidation.
It's like a fire spreading.
Yes. But because vitamin E is structurally anchored right next to those vulnerable lipids, it acts as the ultimate membrane antioxidant. It intercepts the free radical, neutralizing the threat by forming a semi-stable intermediate, and completely stops the chain reaction.
So if a patient lacks vitamin E, their highly active cell membranes literally oxidize and structurally degrade.
Exactly, which is why the primary clinical signs of vitamin E deficiency are profound nerve and muscle degeneration.
Unbelievable. And finally, we have to look at the quintessential water-soluble molecule, which is vitamin C, or ascorbic acid. Mechanistically, it acts as a potent reducing agent and a mandatory co-substrate for specific hydroxylation reactions in the body.
The most critical being collagen.
Yes, the hydroxylation of the amino acids proline and lysine during the synthesis of collagen. By adding a hydroxyl group to those amino acids, vitamin C allows the collagen chains to form tight, highly stable hydrogen bonds.
And collagen is the fundamental structural scaffolding of the human body. It forms the connective tissue, the cartilage, and the structural walls of the blood vessels. Without the hydroxylation step facilitated by vitamin C, the collagen fibers cannot cross-link properly. The triple helix structure is unstable and weak.
It's just inherently defective.
The resulting collagen is fundamentally defective.
Which explains exactly why scurvy looks the way it does. The absolute hallmark of scurvy is capillary fragility. The tiny blood vessels in the gums and under the skin are built with this defective, uncross-linked collagen.
They can't hold up.
No, they simply cannot withstand normal hydrostatic pressure. They rupture spontaneously, leading to petechiae, which are those tiny pinpoint hemorrhages under the skin, and profound bleeding gums. The textbook makes it incredibly clear. No vitamin C means no proline hydroxylation, which means weak collagen, which means your blood vessels physically tear open.
The direct line from cellular chemistry to structural failure is undeniable. But, this mechanistic understanding also forces a critical realization in clinical diagnostics. Deficiencies are not exclusively dietary.
Oh, right.
A patient can consume a perfectly balanced, nutrient-rich diet and still present with a severe vitamin deficiency due to maldigestion, malabsorption, or pharmaceutical interference.
That's a huge point for anyone going into practice. If a patient has an underlying condition, like, say, pancreatitis, their pancreas isn't producing the necessary lipases to break down the food matrix. Without that breakdown, the fat-soluble vitamins remain trapped.
They just passed right through.
Exactly. Similarly, a patient who has undergone a major gastric resection lacks the digestive real estate and the biomechanics to properly emulsify and absorb those lipophilic molecules.
And we also have to be hypervigilant about drug interactions.
Oh, yeah. Like what?
Well, take a patient on cholesteremia. It's a bile acid sequestrant used to aggressively lower cholesterol. By taking it, the patient is literally flushing their bile acids out of their system.
Oh.
And no bile acids means no micelles, no chylomicrons, and a massive drop in the absorption of vitamins A, D, E, and K. We have to treat the whole patient, looking at their surgical history, their pharmacology, and their metabolic state, not just their self-reported diet history.
Assessing the whole patient requires synthesizing everything we've covered today. A clinical learner must hold all of these mechanistic realities in their head simultaneously. You have the strict organic definition of essentiality. You have the cascading progression from a biochemical lesion all the way to structural morbidity.
You have the fundamental divide of solubility.
Right. Solubility dictating transport, storage, and the very real dangers of hypervitaminosis. And you have the specific cellular pathways from retinoic acid signaling to collagen hydroxylation that completely explain the pathogenesis of individual diseases.
That's a lot to hold on to, but it's the foundation of good medicine.
So to help cement that logic before you step onto the wards or, open your books for your next study session, I want to leave you with a quick review exercise that's directly inspired by the textbook's approach.
I love this idea.
Try constructing a diagnostic decision tree for a hypothetical patient. And contrast a suspected vitamin A deficiency against a suspected vitamin D deficiency. But here's the catch. Do not start with the physical symptoms.
Start at the biochemical lesion.
Exactly. Start at the biochemical lesion.
Yeah.
What specific metabolic indicators would separate the visual and epithelial cascade of vitamin A from the calcium and bone mineralization cascade of vitamin D? And how would a patient's history of fat malabsorption complicate your diagnosis of either?
Building that decision tree is exactly how you move from just memorizing facts
Yep.
to actually practicing clinical reasoning.
Yep.
And as you map out those pathways, I encourage you to consider a massive conceptual shift that is currently happening in the field of nutritional science.
The shifting paradigm.
Yeah.
Yeah.
For decades, the entire medical establishment defined nutritional essentiality purely by the prevention of acute deficiency diseases. You give a sailor enough ascorbic acid to prevent his blood vessels from rupturing, and you call the diet a success.
Right. You cure the immediate failure and call it a day.
Exactly. But today, the textbook points toward a shifting paradigm. The modern view of nutritional adequacy is no longer just about avoiding the basement of morbidity. It's focusing on defining dietary requirements based on achieving optimal health, maintaining robust metabolic reserves, and protecting cellular integrity against long-term oxidative stress.
In a modern medical landscape where we have access to abundant, biochemically fortified foods, the core question fundamentally changes. Should clinical medicine remain focused merely on treating the absence of disease, intervening only when the biochemical lesion threatens structural damage? Or is there a newly defined, scientifically rigorous horizon for optimal physiological function that we are really only just beginning to map out?
It's a profound question.
It is. It's a complex question that bridges the gap between basic biochemistry and preventative care, something to deeply ponder as you hit the wards and examine your next patient. Keep digging into the pathways, keep questioning the mechanics, and we'll see you on the next deep dive.